As science advances, we are beginning to see aging not simply as a passive process of decay, but as a complex, regulated biological program, one that shares many underlying mechanisms with chronic disease. Among the most intriguing of these connections lies in the overlap between biological aging and immune system dysregulation, particularly the rise of autoimmune conditions. Emerging evidence suggests that vitamin D, long celebrated for its role in bone health, may operate at a vital intersection, influencing both the aging process and the risk of autoimmune disease through shared molecular and immunological pathways. To understand this connection, we must first look at the nature of biological aging, especially at the cellular level. One of the hallmarks of aging is immunosenescence, a gradual decline in the function and coordination of the immune system. This deterioration includes a reduction in adaptive immunity, especially T-cell diversity, increased chronic low-grade inflammation, inflammaging, and impaired ability to regulate immune responses. These changes not only increase vulnerability to infections, but also create the conditions for autoimmune activity, where the immune system begins mistakenly attacking the body's own tissues. Here's where vitamin D steps in as a central player. Far from being just a nutrient, vitamin D acts as a steroid hormone with wide-reaching effects across nearly every major physiological system. One of its most important roles is in modulating immune function. Vitamin D receptors, VDRs, are present on most immune cells, including T lymphocytes, B cells, macrophages, and dendritic cells, suggesting that this vitamin has a regulatory role in both the innate and adaptive branches of immunity. In autoimmune diseases like multiple sclerosis, rheumatoid arthritis, lupus, and type 1 diabetes, vitamin D deficiency is strikingly common. Not only that, but lower vitamin D levels are often observed long before the clinical onset of these diseases. This suggests a potential causal role, one where insufficient vitamin D may tip immune regulation into dysfunction. Supplementation trials, although still ongoing in some areas, have already shown protective effects, especially in people genetically predisposed to autoimmune conditions. The mechanisms at work here are the same ones relevant to aging: inflammation control, oxidative stress reduction, and regulation of cellular senescence. Vitamin D has been shown to suppress pro-inflammatory cytokines such as IL-6 and TNF-alpha, while promoting anti-inflammatory mediators like IL-10. This balancing act is critical in preventing the chronic overactivation of the immune system seen in both aging and autoimmunity. Moreover, vitamin D influences the expression of over 1,000 genes, including those involved in DNA repair, cell cycle control, and apoptosis, functions that are deeply entwined with both longevity and immune tolerance. In aging, these pathways determine how efficiently cells respond to damage and whether they enter a state of chronic dysfunction, senescence, or renew themselves. In autoimmunity, the same genomic controls determine how immune cells distinguish between self and non-self, and whether potentially self-reactive cells are eliminated or mistakenly allowed to persist. Another fascinating link is found in telomere biology. Telomeres, the protective end caps of our chromosomes, shorten over time with each cell division and in response to inflammation and oxidative stress. Their length is considered a marker of biological age. Studies have shown that individuals with autoimmune diseases often exhibit shortened telomeres, reflecting accelerated cellular aging. Meanwhile, adequate vitamin D levels have been associated with longer telomeres, suggesting that it may help slow both immune dysregulation and the cellular aging process simultaneously. This convergence of evidence leads to a compelling hypothesis. Vitamin D's ability to reduce autoimmune risk may stem from the same anti-aging pathways it influences, particularly those involved in inflammation, DNA stability, and immune surveillance. In this light, autoimmune disease and aging might not be separate phenomena, but rather different expressions of the same underlying biological vulnerabilities, ones that vitamin D is uniquely positioned to address. The implications are profound. For one, it means that maintaining optimal vitamin D levels through sun exposure, diet, or supplementation may offer dual protection, shielding us from premature immune decline while also preserving cellular youth. This is particularly important given that both autoimmune disorders and age-related chronic diseases tend to rise dramatically after midlife. For populations living at high latitudes or spending significant time indoors, as many elderly and chronically ill individuals do, the risk of vitamin D deficiency becomes even more pronounced. In these groups, supplementation of 1,000 to 2,000 IU/day of vitamin D3 is commonly recommended, not just for bones but for total systemic health. Some clinical trials even suggest higher doses may be beneficial in people at risk for autoimmune disease, though personalized monitoring is essential to avoid oversupplementation. In conclusion, vitamin D appears to occupy a unique position in human health, bridging the domains of aging, immune regulation, and disease prevention. By supporting the immune system's ability to distinguish friend from foe, by calming the fires of chronic inflammation, and by protecting the integrity of our genome, vitamin D offers a shared biological pathway for slowing both aging and autoimmunity. It may not be a panacea, but in the ongoing quest for longevity and resilience, this humble nutrient may prove to be one of the most essential.